So the next speaker is Konstantin Senkevich from the Neuro. And he is going, I don't have his title here, but he could give the title. Yeah, sorry about that. I just got so excited about it. Okay.
Okay. So, and my title is the role of other genes in the lysosomal glycosphingolipid metabolism pathway in Parkinson's disease. And by other genes, it basically means other than GBA1. So we all know and well aware about this large GWAS. It nominated 90 SNPs in 78 independent lots. And we wrote a review with Ziv a few years ago and calculated a number of SNPs that played some role in autophagy lysosomal system, including mitophagy, and at least 35 lots played some role in autophagy lysosomal system, makes it quite a prominent number. There is another layer of evidence that the lysosomal genes playing a role in Parkinson's disease. Neuronal and glial accumulation of alpha-synuclein have been shown in human lipidosis. Alpha-synuclein accumulation affecting neurons and glial cells was found in multiple lipidoses like Sandoff disease, Tay-Sachs disease, metachromatic leukodystrophy, beta-galactosidiosis, adrenal leukodystrophy, and Krabbe disease. The main components of lysosomal glycosphingolipid metabolism pathway is demonstrated here. Of course, the pathway is large, but we are focusing on the subset where the central role is playing famous GCase encoded by GBA1 and all the other enzymes around. And during this talk, this figure would be central because I'm going to show each individual enzymatic the role of genes that encode all of these enzymes. So we well aware about risk of GBA1. What about other genes? GALC is our first nominee. So GALC was nominated in Parkinson's disease GWAS as one of the hits. And recently we demonstrated that Parkinson's disease GWAS top hit in GALC is also strongly associated with increased GALC activity. That's a bit different from what we expect in terms of our knowledge on GBA, as the activity of GK is decreased in Parkinson's disease. Here, relationship is a bit more complicated. We also demonstrated that by colocalization that this SNP is also increased GALC expression, and Mendelian randomization point to the similar direction that increased GALC activity could be causally linked to Parkinson's disease. The relationship between GKs and GALC could be a bit more complex. We found that there is a strong correlation between GKs and GALC in homozygous carriers, and it reduced in heterozygous and absent in non-carriers, almost absent. Therefore, there could be a complex link that increased GALC activity interact with GKs and therefore risk of Parkinson's disease. Another nominee is ARSA, this gene dysfunction of which causes metachromatic leukodystrophy, and it is again in this pathway. So, first paper was published in 2019 in which they performed co-segregation of a family with several Parkinson's disease patients. They found that indeed this variant L300S co-segregated into Parkinson's disease patients. They provided some functional evidence that ARSA is an alpha-synuclein chaperone, but I'm focusing here only on genetic evidence, therefore I would not discuss this in details. They also provided some evidence on the association of common variants. This was not replicated after. We summarized all the evidence on ARSA from the genetic standpoint in the recently published paper. So if we track the efforts to perform burden analysis for ARSA, smaller studies did not find any signal in ARSA, and larger studies found some signal. So originally it was found the prevalence of loss of functional variants in late onset Parkinson's disease. It's relatively small, therefore it was not statistically significant. But two more recent studies by Mary Macarius from NIH and ourselves found that there is a burden of rare loss of function variants among Parkinson's disease patients. So overall signal suggests that there is a burden of rare loss of function variants, and it was replicated already several times. What about non-synonymous variants? We also attempted to co-segregate families with Parkinson's disease. So we contacted lysosomal storage disorder center. We found 10 families with metachromatic leukodystrophy and sent them questionnaire whether they have a relative with Parkinson's disease. Two families contacted us back, and we co-segregated this variant, but this patient was also a carrier of GBA1 recombination. And in another family, another Parkinson's disease patient was obligate carrier, but he was diseased, therefore he was not able to test, but his pulse was wild type, therefore he should be a carrier. And other branch of family carried different variant, and we did not segregate. So we have a quite weak evidence, but suggestive for that this variant that disrupt ARSA activity could be also a risk variant. But this should be replicated as well as other variant L300S. So next gene that we nominate is GLA. GLA is also in the pathway, and there is no strong genetic evidence about this gene, but several studies, one led by Dr. Alcalay and another from Portugal, they studied prevalence of Parkinson's disease among families with Fabry disease, and they indeed found that prevalence is higher among these families. In a study led by Dr. Alcalay, the prevalence was extremely high, actually, more than 8% in the age older than 60, and quite a high family history percentage. Another independent study from Portugal also found overrepresentation of Parkinson's disease patients in the families, and it was statistically significant, so it was higher than in general Portuguese population. Another way to link Parkinson's disease to alpha-galactosidase A activity, it was found that overall activity is lower in Parkinson's disease cases, and it was already found in two independent studies, again by Dr. Alcalay, and another study that was very recently published. And higher activities could be linked to lower odds of Parkinson's disease. This study that was recently published in Movement Disorders by Miller et al., they found that variant A313T has higher frequency in Parkinson's disease cases versus controls, but they did not statistical significance, so they meta-analyzed with another available study from Slovakia, and that was a small study. After meta-analyzing two studies, they found statistical significance for these specific variants. Our preliminary results does not replicate this, but we're still working on it. It's too early to demonstrate it. So I'm not going to stop on SMPD1 after this beautiful talk, but I have to mention it here. I'm just repeating 25 minutes talk in 5 seconds to show that, yes, that's most probably Parkinson's disease gene, as was shown originally by Ziv and then replicated in this large-scale study. So overall, all this pathway turns green. We have extreme enrichment of variants and genes that have at least some risk of Parkinson's disease. Obviously, that's GBA1, but all the other genes and enzymes that they encode seems to be playing at least some role in the disease. But this, of course, is a piece of the pathway, of sphingolipid pathway, and overall, it's a subset of all lysosomal genes. As it was previously mentioned, several efforts tried to study all the lysosomal storage disorder genes together, and the study led by Joshua Schurman and Lori Robach demonstrated that there is a burden of lysosomal variants in Parkinson's disease even after excluding GBA, and they pinpointed several genes, including ACH1, that is also from sphingolipid pathway, and cathepsin D. More recent study tried to replicate similar analysis, studying on a smaller sample size. They did a burden of 50 genes linked to lysosomal storage disorder, and indeed they found a burden of all lysosomal storage disorder variants in Parkinson's disease patients as compared to controls, but they failed to replicate results for sphingolipids. They found enrichment of mucopolysaccharidosis genes in Parkinson's disease. So in our study, we also performed we sequenced 36 lysosomal genes from cohorts available at McGill University. And we also included UKB bank and MPPD and a total of more than 8,000 patients and 68,000 controls. That's the genes that include in this study. We did not include in this specific study GBA1, GALC, or ARSA, since it was already well published and demonstrated as the cohorts that we include: Columbia, Sheba, McGill University, and Pavlov human brain cohort, as well as UKB and MPPD. So we performed burden analysis of different genes, stratifying by all rare variants, variants non-synonymous, and variants with high deleteriousess defined as a loss of function or by a CAT score. We also performed analysis stratifying by different domains, and we found that one of the missing points, hex A, in one of the domains linked to Parkinson's disease, as well as ST3, GL3, all rare variants linked to Parkinson's disease. This p-value survived multiple correction for number of genes. And we also found similar results as in analysis by Robuck and Joshua Schulman, that ACH1 linked to Parkinson's disease nominally. Therefore, this potentially, since it's replicated now in a couple of studies, suggests that there is some signal, but it's too weak to be significant in a smaller sample sizes. So overall, we have some suggestive evidence for most of the enzymes and respectively genes that they encoded. We suggest that there is a strong enrichment for glycosphingolipid metabolism genes and also glucosamine-glycan metabolism genes. So it's basically pathway analysis of genes involved in the burden as a result of our burden analysis. And quite a few genes involved in the glucosamine-glycan pathway, and not only glycosphingolipid pathway. And obviously, if we include genes that have been proven earlier with a link to Parkinson's disease, the significance increase. So, to the conclusion, there is a strong role for genes other than GBA1 within the lysosomal glycosphingolipid metabolism pathway in Parkinson's disease. The interaction among different components of the pathway should be carefully studied because it could be much more complex, and even that GBA1 playing a major role, the role can be modified by all the SNPs and all the genes involved in the pathway, and we need to better understand this to define best targets for drug development. I would like to thank all the collaborators, all the funding agencies, and of course our amazing club, Dr. Ganur. Feel free to reach me out with any questions or collaborative suggestions. Yeah, sure.
Great talk. I'm curious for the GAL-C link. What's your hypothesis for how increased GAL-C activity might contribute to PD? And have you looked in some PPMI databases to see, do you see evidence of GAL-SARE, GAL-SPHING accumulate in variant carriers?
Yeah, thank you for the question. So in our study, we did some functional analysis, and in the knockout of alpha-synuclein, we did not find, in knockout of GALC, we did not find accumulation of alpha-synuclein. This is slightly contrary to the data that in the Krabbe disease of infants, there is some accumulation of alpha-synuclein. So the link is a little bit controversial. It could be, since the GALC is very close to GCase, that there is a complex interaction with the increased activity of GALC. There is some change in the activity of GCase that compensatory decrease. But these complex interactions need to be studied much more carefully to understand the link.
Thank you.
The only enzyme in your pathway that is X-linked is alpha-galactosidase A. How did you account for that?
Thank you. So we did not yet study ourselves GLA. That's work in progress. But in the study that is ongoing. We separate males, females, and on meta-analysis we apply additional package to account for this.
They don't have Fabry disease.
No.
Konstantin, this is wonderful. Thanks for doing this. I'm curious about common variants and thinking about ways to analyze common and rare variants together at each of these genes. We've actually done a study looking at GWAS data, summary data from the largest studies. It's actually buried in a kind of functional paper, so many may not be aware of it. But we found that there is a common variant burden across these genes as well in association. And for example, at ARSA, there's a strong suggestive signal. We've talked a little bit about it. So I think it would be very interesting to consider both rare and common variants together in an analysis, although I know that's easier said than done.
Thank you for the question. Actually, there is a scuttle package to account for this, so it could be quite easily done, from what I know, to try to account for both common and rare statistically. Yeah, that could be interesting, but from what we are trying to add to this paper is burden of PRS, so basically polygenic risk score in independent cohorts of same genes. So far, studying only sphingolipid closed pathway, we did not find any important PRS signal. There was a poster yesterday, and I'm trying to exchange set of genes because they found a very strong signal for 100 lysosomal genes. So I'm just curious what is driving it and if it would be significant after excluding GBA1.